The portals of the Knowledge Portal Network for type 2 diabetes, cardiovascular disease, cerebrovascular disease, and sleep disorders aggregate, analyze, and display genetic and genomic results with the aim of helping researchers better understand these complex diseases. This video will take you through the variant page, one of the core elements of the Knowledge Portals. We'll demonstrate using the Type 2 Diabetes Knowledge Portal, but the page looks the same in the other portals. To see a variant page, enter a variant ID into the search box on the Knowledge Portal homepage, or click on a variant ID anywhere it appears in the portal. The top of the variant page summarizes basic information about it: its location, the reference and variant alleles, the transcripts in which it's located, the impact of the variant on the gene where it's located, and predictions of how deleterious it might be, generated by the polyphen and SIFT algorithms. The Associations at a Glance section uses two kinds of plot to summarize associations for this variant across a wide range of phenotypes. First, the PheWAS plot shows significance, the negative log of p-value, on the y-axis versus phenotypes on the x-axis. Phenotypes are color-coded by their category, for example, glycemic or cardiovascular. The default view shows the most significant association for each phenotype from the datasets aggregated in the portal. Click on a data point to see details. Using the radio buttons at the right, you can choose to view bottom-line analysis, the results of a meta-analysis that takes into account sample overlap between datasets, performed across all datasets in the portal. Another option displays a point showing the phenotypic associations from each dataset included in the portal. The final option generates a plot for a PheWAS analysis performed at the University of Michigan that shows associations for this variant across a wide range of phenotypes, drawn from UK Biobank data. The second kind of plot shown in this section is a forest plot. This plot shows associations for the variant across all phenotypes in the portal, color-coded by significance. To see the legend, mouse over the upper left-hand corner of the table. The x-axis of the plot shows the beta, or effect size, of the association, and each association shows horizontal bars indicating the size of the confidence interval. The Associations at a Glance section gives you an overview, but what if you want to drill down to the fine details? The next section, Associations across all datasets, lets you do that. It includes complete information on all the associations of this variant in the datasets of the portal. The associations are plotted in a graphic that shows significance versus dataset sample size, and each association is listed in the table below. Because there are so many datasets in the portal, this can be a little overwhelming at first, but it's very easy to filter the display to see the associations that you're interested in. Start typing the name of a phenotype and select one, or choose a trait group. This filters both the graphic and the table, so the only selected phenotypes are shown. You can also filter the plot by entering a range of p-value or sample size. Mousing over a point gives complete details for the association. In the table, the most significant association for a phenotype is shown first. Click on the Expand icon to see all datasets. P-values in the table are highlighted green to indicate their significance. Moving further down the variant page, the Epigenomic Annotations section summarizes the tissue-specific chromatin states that overlap the position of this variant. They can be viewed and filtered in the table and are illustrated in two different summary graphics below. The next two sections, Genetic Association Interactive Tool and Locusome, need more in-depth explanation, and we'll focus on them in separate videos. Finally, the How Common Is section shows minor allele frequency for this variant in different ancestries, seen in the single ancestry datasets included in the portal. The datasets with the largest sample size are shown by default, but you can click on the Show All Cohorts button to see every dataset. At the bottom of the variant page are links to pages specific to this variant in selected external resources. Please let us know if there are any other links that you would find useful.